Thank you very much. Tēnā koutou. Thank you very much for the welcome this morning. It's much appreciated. Ko Mokatero te maunga, ko Rakahuri te awa, ko Ngāti Pākehā te iwi, ko Takuingoa Andrea Byrom. I am the whakahaere, or the director of the New Zealand's Biological Heritage National Science Challenge. And I'm actually going to talk about science this morning, but don't be alarmed. I'm actually going to show you some actual data and graphs, but it's really easy to understand and hopefully also will inspire you. So, it was really interesting. I got given this fantastic talk title called The Role of Large Projects and the Importance of Research, and I thought, what does that mean? I bet Campbell wrote that. Did you? Yeah, you did. I knew that. I could tell. So I thought, right, what am I going to talk about? And kind of talk about the National Science Challenge and what it actually is in a way that all of you can appreciate and understand. And then also I am going to talk a little bit about what Te Reira just mentioned at the end, which is how do we learn what we're achieving in these big, large-scale transformative projects in such a way that we can contribute to a national conversation. And I would really like to acknowledge a postdoc who's been working with us at Manaaki Whenua Landcare Research on that topic. So I'm kind of wearing two hats, as a scientist and then also as a director of the Science Challenge. And I'll kind of bring it back out to the big picture and what I think science and research can contribute to this part of the overall conversation in these fantastic transformative projects. So very briefly, the New Zealand's Biological Heritage National Science Challenge was established in 2014. We have an amount of funding over 10 years that amounts to about $63 million. And one of the cornerstones of our work is to work in partnership with Māori at every level of the challenge, from governance right on down to communities and everything in between. Until very recently, our management team that I work on, I'm the director, I've got a management team with me. 50% of us were Māori. We've had one person step back into their research, so she's kind of been fledged to work back in the science community again, which is fantastic. We're also 50% women, which is really good as well. A lot of what we're doing is working with central, local government, business, philanthropic organisations and so on. So it's really about doing science in a much wider context. These are all of our Science Challenge parties or collaborating partners in what we're doing. So we've got every university and every Crown Research Institute in the country signed up to our kaupapa, if you like. We've also got the Department of Conservation and the MPI, Ministry for Primary Industries, as well, in recognition that they hold science expertise in those organisations. And we've got more wanting to jump on board in the next little while. Rude, I think it was, mentioned this morning about, what did you say? I wrote down this quote from you. Split the DSIR into 15 competing organisations, and I laughed and I thought, yeah, well, now what they're doing is trying to kind of stitch them all back up again, except that this is like the DSIR on steroids because we've got all the universities involved as well. And so trying to break down those barriers between the silos that have built up in science. And if that sounds familiar to you, it should be, because that's exactly what we're trying to do nationally. It's what all the speakers have talked about this morning around collaboration and breaking down those barriers. And that's exactly what we're trying to do in our Science Challenge, is to break down those barriers between all these organisations, stitch people together, get them joined up again in such a way that we can actually help properly with these national big picture projects. So our mission is that. I'll leave you to read that. But again, as I said before, it's really about doing science in a much wider context, involving Māori, involving communities, involving international collaborators as well. So a much bigger collective than we've had in the past. It's not about scientists squirreled away in their labs, not telling the world what they're doing. So that's enough on our Science Challenge for now, except to say that one thing that we've done as part of that wider context around bringing making it real, I guess, is to have flagship sites. And one of our flagship sites for our Science Challenge, and the reason that we were so happy to sponsor this conference, is because Cape City is one of our flagship sites. And that's in recognition that there's a lot of amazing research being done here, primarily by Manaaki Whenua Landcare Research. And I forgot to mention that Manaaki Whenua Landcare Research are our host organisation for our Science Challenge. So we're actually not another competing entity in the New Zealand science system. We do receive funding, but we disperse it back out to all of our challenge parties. We don't compete for extra funding in the science system. And so that was the idea, is that Manaaki Whenua Landcare Research would host our Science Challenge, and they've been a fantastic and very supportive host. So anyway, we've got these flagship sites, and that's in recognition, again, that science and research plays a role in these big transformative projects. And we liked Cape City because of the transformational vision and the drive towards multiple outcomes. So for example, retaining that primary productivity in a sheep and beef farming and wine growing landscape, in addition to our natural ecosystems, that was really appealing to us, in addition to the social and the cultural desired impacts and outcomes. So it's a really transformational project. And of course, the big question that's in my mind as a scientist and a director of a science challenge is then how does that connect up to a whole bunch of other things that are going on around the country, and what can we learn from all of them? So there's all sorts of stuff going on, some of which you're yet to hear about, and some of which we've already heard about this morning. And I guess the point is that if we can learn something from all of this, then we can really start to make progress. And what I really like to think of it as is a bit of a feedback loop. So as we learn more, we are supposed to be feeding it back to you and giving you the evidence to support your work. So that's what I'm going to do next, is now divert onto speaking about some of the research that's being done at Manaaki Whenua by this wonderful woman called Rochelle Burnie, who couldn't be here today. So our host, Manaaki Whenua Landcare Research, they actually, because I've been seconded into this role in the Science Challenge, they funded a postdoctoral fellow, who is Rochelle, to work with me to keep some of my own science and research activities going. And she's been doing some really nice work that I want to tell you about now, and then I'll bring it back out to that bigger picture just at the end of my talk. And then also I'd like to acknowledge our colleague John Innes, who also couldn't be here today, and he's been working with me and Rochelle. We have another colleague working with us who is here in the audience, that's Roger Peck. Roger's now retired, so we don't put the photos of our retired scientists on our slides anymore. I'm joking. I just didn't have a photo of Roger, but he's sitting there in the red jacket halfway down the aisle if you want to talk to him at lunchtime or whatever. So what I'm going to do is I'm going to present the results of what Rochelle's been looking at on a scale like this. So all you have to do is remember this once. Down the bottom here, what Rochelle's been doing is she's been asking the question, how successful have we been and what kind of biodiversity responses have we had when we look at sanctuaries and really large-scale predator control sites around the country. And what she's done, and I'm not going to go into this, but she's gone out to DOC and she's gone out to Sanctuaries of New Zealand, and a whole bunch of people have thrown a whole lot of data at her. And for those of you that are in the audience who religiously keep your spreadsheets with the numbers of predators you've caught and the number of kaka you've counted and the number of bellbirds that people have in their backyards, that's the kind of data we're talking about. Rochelle has spent hours and hours and hours and hours of her life collating all that kind of data and information into a massive database. She's actually got 800,000 records in the database now from a bunch of sanctuaries and mainland island sites from DOC, from all over the country, mostly forest sites, but not entirely forest sites. And the way that she can analyse those data in a comparable form is to do what's called a meta-analysis. And all it means is that each of those measurements, for example, we might be measuring bellbirds before and after pest control. And you might have 10 bellbirds at the beginning and 100 bellbirds at the end. And the difference in the number is called an effect size. Did we have a positive effect? Yes. That goes into this graph as an effect size. So on the right-hand side here, we've got a positive effect. If there was no response of whatever the biodiversity thing was to pest control, then it sits on the line here at 0. And if we've got a negative response, then it's below the line or to the left-hand side. So a positive response might be something like an increase in abundance of bellbirds or New Zealand dotterel, which is what this is, with a little chick right there, if you can see him, her. Or nesting success, or counts per unit search effort, or browsing on foliage. Anything and everything goes into the same analysis, and that means that you can compare anything and everything. It's like being able to compare apples and oranges together. So I'm going to show you a few of these plots. Bear with me, they get more exciting than this one. Because of the amount of data and information that you all know is collected out there, Rochelle's had to choose a couple of things to kick off. So she's chosen fence sanctuaries, and she's also chosen DOC's mainland islands. What she hasn't got yet is other data from places like Cape to City or from our offshore islands, but those will be the next steps, of course. So, and her question is, how do New Zealand's flora and fauna respond to pest control? And if we understand those biodiversity benefits, then we can start to understand where are we fitting in that predator-free big picture that Ed talked about this morning. So I'm going to skip straight to her results now and tell you the good stuff. So at the top here, we've got these little symbols. So we've got birds, native birds. Little fantail represents the native birds. This thing here, which looks like some kind of thrush or starling, that represents the introduced birds. And then we've got flora, represented by the rata flower, purakawa flower, sorry, and that's mostly native flora. And then also this thing representing invertebrates. So you're going to see the same little symbols again and again as well.
It's not a thing.
It's just a bug to me, Rude, you know? It's either a positive or negative on a grid, just reducing it down to the bare bones. Anyway, so the big question, does pest control have a positive effect on native flora and fauna? Yes, we all knew that already, but it's really interesting to put this information on these kinds of graphs. So these are all of her 800,000 records now, captured in all these sanctuaries, fenced and unfenced. And then what she's done is she's split them out into the fenced sanctuaries, how much of a positive effect do they have, and the unfenced or mainland islands, how much of an effect do they have. And as you can see, generally the fenced sanctuaries where we're removing all the pests are doing better than the mainland islands. That is absolutely no criticism of the good work that DOC does. It just means that, again, we all know that it's really hard to deal with reinvasion. And that's why these landscape scale projects and getting bigger and bigger and bigger is so important. So that's interesting. We've got all the sanctuaries, all the fenced sanctuaries and all the mainland islands. Those three colours you're going to see again and again and again. So here we go. If you start splitting it into these taxonomic groups, or species by species if you like. So we've got the flora at the bottom, the wonderful native invertebrates. In the middle, birds lumped together at this stage, in the top, middle top, and then at the very top is all of those things combined. And so as you can see at every level, also what Rochelle has discovered is that we're starting to have a positive effect. And we want to know that as well, because we want to know that we're having an effect on our communities, ecological communities, not just on perhaps one or two bird species. So that's neat. And now it gets even more interesting, because what Rochelle has also done is then split that into fenced and unfenced sanctuaries. And so on the red is the fenced sanctuaries and the green is the unfenced sanctuaries. And you can start to see, for example, that birds do pretty well in the fenced sanctuaries. We all knew that, but now we've got some numbers on it, not just a sort of a gut feeling. Not so great though in the unfenced mainland islands. And likewise, for example, when you look at all the flora and fauna together, it's a little bit questionable as to how much of an effect we're having on New Zealand's native biodiversity. So we keep going. The next thing that she's drilled down into is just to look at the birds. So again, generally a positive effect on birds overall, so these are all birds lumped together. But interestingly, our native birds are doing better than introduced birds when we remove pests, whether they're in fenced or unfenced sanctuaries. So that's a bit interesting too. Great, we're having a positive effect on our native flora and fauna, more so than on our introduced. That's another big win for the good guys. And then if you split it up again, you get some interesting trends. So again, those fenced sanctuaries are doing better for all birds and for our native birds. But interestingly, the introduced birds, and also for the introduced birds. Okay, so you might start to ask the question, well, what does that mean in terms of the different components of the community? So the next thing that Rochelle's looked at is guilds of birds. And these are just, guild is another word for functional groups. So different things eat fruit, that's the frugivores. Different things eat beautiful native invertebrates. I was going to say bugs, but I didn't. Things like riflemen's and tomtits are insectivores, and so on. We've got our omnivorous species and our carnivores, our top predators. And that's pretty interesting too. So what you can see is that generally we're having a positive effect on all of our native birds, except for our predatory species like the kahu or the New Zealand falcon. Now, in a way, that's very interesting. If you're trying to restore ecosystems internationally, there's a huge debate in the literature about bringing back the top predators. So suddenly it also puts another layer on the whole, quote, predator-free New Zealand concept. Because we actually don't want to be predator-free, we actually just want to be introduced predator-free. We actually want to bring back, eventually, our top predator species. And kingfishers are a predator. I looked it up the other day. They eat lizards and other things. So that's pretty cool. We can start to tease out what's happening at a community, ecological community level. And again, what Rochelle's done is split that out by the fenced and the unfenced sanctuaries. So again, with most of these guilds or functional groups of birds, they're doing better in the fenced sanctuaries. It means we've got to work really hard in our unfenced areas. That's why we need transformational scaled up projects like Cape to City. Somebody else already alluded to the PCE's report. The Parliamentary Commissioner for the Environment, Jan Wright, before she retired, produced this fantastic report, and I read this thing cover to cover. It is an amazing thing, and if you haven't had a good read of it, I recommend you do. It's downloadable for free from the Parliamentary Commissioner's website. It's really neat. It kind of like, maybe it just spoke to me too much because it's everything that we in science have been trying to do for the last, 25 years or more. But anyway, one of the things that Jan mentioned was this issue of endemism. And so she's kind of characterised New Zealand birds in this way that I'd never seen done before, even though we all know that kiwi are our deep endemics. And so in terms of species, genus, family and order, for those of you that know, species are pretty generic worldwide, or at least they might be unique to New Zealand, but there may be other species in that particular genus that are in Australia or somewhere. But the deeper we go, New Zealand is obviously the only place that has kiwi. And that was an interesting way of characterising that. And so what Rochelle did was she looked at the endemism responses of she looked at the responses of the different bird species by endemism. And she said that when she found this, she was sitting in her flat with her flatmates all around her, and she was leaping out of her chair with excitement. And they thought that they looked at her like she had two heads, because they couldn't understand why she was so excited. But this is really interesting because our deep endemics are responding incredibly strongly to predator control. So good on you, all you good people out there. And likewise, we see the same thing with the fenced and the unfenced sanctuaries. And I should mention, if there isn't a red dot here, it just means that we didn't have any data for those particular places. I am coming to the end. I've got a 5-minute thing. What's really important about this is that Rochelle's been able to do this. If we split this out by species, there's a whole pile of species that we can look at that are somewhere on this graph from negative to positive in terms of their responses. Some of the more introduced ones are down the bottom, but then look at things like the eastern rosella do quite well under predator control. Those are the things that Bruce was worried about getting into his fruit. And if you heard me and Bruce on the radio last week, That was pretty funny. And then the same thing, we can split that out by the fenced and the unfenced sanctuaries. So I just want to bring it back out to the big picture briefly, just to finish off. The science challenges are really about big collaborations, combining our infrastructure and comparing that, national expertise, collaborations, using the citizen science, that should be obvious from this work that we've done, and working in partnership with Māori. We're also paying a lot of attention to how we can, as a science challenge, do some work, like what Rochelle's done, that helps deliver on big visions like predator-free or biosecurity 2025. There's lots of freshwater ecosystems in trouble, we all know that. And so we've got some long-term visions that we're trying to deliver on as well. And I think these are just a few of our other projects that we're working on that I won't go into. Breakthrough tools for predator-free, working closely with Ed and his team. New tools for conservation, genomics, building resilient, connected habitats across New Zealand. And that, as I mentioned before, is where these large-scale projects really come in. You guys have a national context. We want to learn from you and from all of these other projects. And I think in that context, science and research has a really crucial role to play to help us understand what we are delivering for New Zealand. So thanks.